Welcome to my channel. Today I will discuss about conditionally essential amino acid. First, let us know what is conditionally essential amino acid. Conditionally essential amino acid actually, or mammals, or we can produce this or we can convert this, but these are not enough due to some physiopathological condition, especially in case of infancy, trauma, or some other stress or growing condition. Then we need this from the supply. And there are 6 amino acid which we call conditionally essential amino acid. Let's discuss about this. First, arginine. In mammals, arginine is the precursor of nitric oxide. How it produces? Nitric oxide can convert to L-arginine can convert to nitric oxide, which has several functions like vasodilation, which actually enlarges the blood cell, and then neurotransmission, immune response, and wound healing. And it has also a great impact on urea cycle function, because arginine is a key intermediate in urea cycle which helps to detoxify ammonia from liver. In plants, when arginine becomes conditionally essential in plants during seedling development and when plants face mechanical wounding or pathogen attack or different type of abiotic stresses. From the structure we have seen that arginine contains a lot of nitrogen, and this group known as guanidine group. So arginine is a big storage of nitrogen and also transport. In the weight basis, arginine contains 32.2% nitrogen. And again, arginine is a precursor of polyamines like spermidine, spermine, which can contribute in cell division, elongation, and some other biochemical function. It also helps to scavenge ROS, reactive oxygen species. At the same time, it has a great impact on stress tolerance. Again, arginine is a precursor of nitric oxide. In plants, nitric oxide acts as a signaling molecule. It helps to give the signal in case of root development and elongation, stomatal opening and closing, and also plant stress, and give that defense from different abiotic stresses. In case of fish, arginine is considered as an essential amino acid because in early development stage they can't produce. And in case of cats, arginine is considered as an absolutely essential amino acid, and it must be supplied in daily basis, otherwise cats can die due to hyperuria. And in case of birds, same thing. These are arginine considered as an essential amino acid. What about cysteine? Cysteine is synthesized from methionine via transsulfuration pathway, and in mammals, cysteine is a precursor of glutathione, a major antioxidant metabolites, which has a great impact on antioxidant defense. As we know that antioxidant defense system is consist of ascorbate and glutathione pathway, and also it helps to detoxify, supports liver detoxification process, and most importantly it contains disulfide bond. Cysteine and cysteine can form disulfide bond, which helps to stabilize protein tertiary and quaternary structure. What about plants? In case of plants, cysteine become conditionally essential amino acid under oxidative stress and obviously during sulfur deficiency. And it has a big function in plants. Again, it's a precursor of glutathione and also secondary metabolites glucosinolate, which helps defense plants from different herbivores, pathogen and heavy metal detoxification. Cysteine rich peptide like phytochelatine chelate heavy metal and then it detoxified. And again, cysteine is a precursor of another signaling molecules, hydrogen sulfide. These signaling molecules have great impact on plant growth and stress response. It also helps to maintain redox homeostasis. So let's talk about oxidative stress. In plants or in mammals, usually our due to cellular mechanism, a lot of ROS can generate. But at the same time, our oxidative system, our antioxidant system can scavenge this this generated ROS. But when ROS generation is excess, then the antioxidant defense system, then plants and mammals face oxidative stress. That means an imbalance between the production of ROS, free radicals, and the ability to neutralize them with antioxidant. Look at the glutathione structure. We have seen that glutathione contain thiol groups. These thiol groups come from the sulfur, and this glutathione along with ascorbate can detoxify ROS through a cycle, non-ascorbate glutathione cycle. Here, glutathione convert oxidized glutathione by the activity of DHAR, and it convert into ascorbate. It helps to convert from DHA to ascorbate, and then ascorbate convert to DHA with the help of APX, ascorbate peroxidase, and this APX helps to detoxify ROS into not toxic water. By this ascorbate glutathione pathway, plants and animals can detoxify ROS. So now I can discuss about phytochelatin. Phytochelatin, these are actually peptide metal binding ligands, and they are repeating of glutamate and cysteine, and as a terminal, third one is glycine. So this glutamine, cysteine and glycine can produce phytochelatin, and these phytochelatin along with glutathione can bind with heavy metal, and then they transfer it into the cell vacuole through this transporter, and in cell vacuole this heavy metal and phytochelatin complex can degrade it and make it non-toxic or normal metabolites or normal metals. Then in case of glutamine, in plants, glutamine is very important because it helps to nitrogen assimilation. If we look at the GS-GOGAT cycle, here GS stands for glutamine synthetase and GOGAT stands for glutamate synthase. This GS-GOGAT pathway is one of the vital pathway to assimilate nitrogen. Look at here, nitrogen uptake as nitrate, and due to the activity of nitrate reductase and nitrite reductase is converted in ammonia, and also plants can uptake ammonia, and this ammonia ion then mix with glutamate, and with the activity of glutamine synthase, cytosolic glutamine synthase, it convert into glutamine, and then this glutamine export in root or shoot in other part. At the same time, due to photorespiration and other biochemical cellular activities, it produce ammonium, and this ammonium then mix with glutamate, and then in plastic glutamine synthase, with the activity of glutamine synthase, it convert glutamine, and again this glutamine mix with oxoglutarate and then produce glutamate with the help of GOGAT. Again GOGAT stands for glutamate synthase, and then this glutamate can pass into the cytosol, and then again it form the glutamine, and in some case it can store as glutamate. So nitrogen can be transported as glutamine and can store as glutamate. And when glutamine become conditionally essential in plants, of course during nitrogen assimilation and also recovery from abiotic stress. As I discussed earlier, that glutamine has a great impact on nitrogen storage and transport, and these are the donor. This glutamine is a donor, nitrogen donor for the synthesis of other amino acids, nucleotides, and also chlorophyll. At the same time, glutamine can have a great impact in osmotic adjustment during different abiotic stress like drought, salinity, and pathogen attack. Now I will discuss about glycine. When glycine become conditionally essential for plants, of course under stress condition and also during active growth and development, when required use purine, chlorine, and heme, and most importantly when plants switch its nutrient source from inorganic to organic, then glycine become conditionally essential for plants. Role of glycine in plants. First, photorespiration, because glycine is a key intermediate in photorespiration cycle, and again this glycine can join with glutamate and cysteine to form glutathione. As I told earlier, that glutathione is a very big antioxidant metabolites which can detoxify ROS. And again it's one carbon. In case of one carbon metabolism, a glycine donate one carbon unit for folate mediated metabolism, which is essential for nucleotide and amino acid synthesis. And in case of defense, there are a lot of review and lot of research there that exogenous glycine application can improve plant tolerance to salinity, drought, heavy metal, and other abiotic stress. And most importantly, glycine can chelate with different type of micronutrient like iron and zinc. These chelate help to improve the nutrient uptake and mobility inside the plants. Let's talk about the proline. Proline has a big impact in case of amino acid, in case of protein structure formation. Especially it causes kinks and also turns and loops. This proline, due to its cyclic structure and different type of structure, rigid structure, it helps to form turn and loops. And in plants, proline also perform a lot of functions like maintain NADP plus pool, and of course protein synthesis, and it function as osmolyte. I will discuss later what is actually osmolyte is. And also it can contribute to maintain redox balance, and also it has a big impact on growth and development. So let's talk about osmolytes. Osmolytes helps to maintain volume and function of cell, particularly under stress, and they help to maintain the fluid level. From extracellular fluid it can come inside, and from inside it can go outside, so it keep to maintain the volume and function of cell. Here in case of native protein urea, as we know, urea is denaturing agent, can form unfolded protein, and then if we apply osmolytes in, again come back the native shape. So like amino acid, proline and glycine play vital role to as a osmolytes. Let's talk about tyrosine. In plants, tyrosine become conditionally essential when pathogen attack and also UV exposure. From the Shigimate pathway, we know that tyrosine, tryptophan, and phenylalanine produced in plants due to the pathway of Shigimate pathway. Through Shigimate pathway, in Shigimate pathway, phosphoenol pyruvate and erythros 4-phosphate can go through Shigimate pathway and form tyrosine, phenylalanine, and tyrosine also can convert it from phenylalanine, and then this tyrosine can further produce different type of secondary metabolites. And again This tyrosine derived metabolite tyramine. Tyramine is a important defense in important metabolites during defense mechanisms. In human, tyrosine helps to produce melanin, thyroxine, and catecholamine. And as we know, tyrosine can convert in melanin, and this melanin is responsible for the pigment in skin, hair, and eye color. And due to the mutation of phenylalanine hydroxylase enzyme, as I told earlier, that tyrosine converted from phenylalanine through the enzyme phenylalanine hydroxylase. So due to the mutation of this enzyme, phenylalanine can't convert into tyrosine, and then phenylketonuria disease can happen. PKU disease, we know it. PKU disease. Also, albinism occurred due to the lack of an enzyme named tyrosinase enzyme, which can't convert tyrosine. So let's talk about the summary. Conditionally essential amino acid in human, also in case of plants, it become conditionally essential because plants or animal can produce this amino acid, but these are insufficient due to some physiopathological condition like stress, growing condition, or other condition. There are 6 amino acids we can consider it conditionally essential amino acid. First, arginine, which is a precursor of nitric oxide in both mammals and plants. And also we have seen that in case of plants, arginine is a big player of nitrogen storage and transport because it contains 32% nitrogen by weight. And cysteine is a precursor of glutathione, which is an antioxidant metabolites, and also precursor of another signaling molecules, hydrogen sulfide. And also cysteine can form phytochelatin, which help to detoxify heavy metals. And in case of glutamine is considered as the nitrogen transport and storage through GS-Gogurt cycle, and also serve as nitrogen donor to other amino acid, nucleotide, and chlorophyll. And glycine combine with glutamate and cysteine to form glutathione, and again glycine chelates helps to uptake different micronutrient and also transport of these nutrients. And proline play a big role in case of structural is a protein structure. It has a big role especially in turn and loop, and it also act as osmolyte. And what about tyrosine? Last one. Tyrosine is a precursor of phenolic compounds and alkaloids, and again tyrosine is converted into melanin, and PKU disease occur due to the mutation of phenylalanine hydroxylase enzyme. Thank you very much. Let's talk about limiting amino acid. What is limiting amino acid? Limiting amino acid, these are essential amino acid. That means these must come from the diet, and then due to the shortest supply of these essential amino acid relative to body requirements, for maximum protein synthesis halter. That means maximum protein synthesis can't occur due to the shortest supply of these essential amino acid. Just look at the picture here. Here in first picture we have seen that lysine is limited here, but protein can synthesize up to this mark, but due to the lack of the lysines, our body can't produce this much protein. Again, if we supplement lysine up to this mark, then our body can synthesize protein up to this mark. Again, in another picture, we have to make car, C here, car with this C, A and R. Here there is 5C and 7A and 6R. So let's make car. So we can make 5 car. Although why? Because we have only 5C. and again we can't make more because R is only 1. That means first limiting is C and second limiting is R. So if one essential amino acid is significantly lower in quantity than others in a protein source, and it becomes the limiting factor for that protein synthesis. Let's have some idea about different cereals. In cereal grains, lysine is limiting. That means the lysine isn't enough there. In case of legumes, methionine first limiting and tryptophan act as second limiting. And vegetables, methionine first limiting and lysine second limiting. Potato, lysine first limiting amino acid and threonine second limiting amino acid. In maize, lysine first limiting and tryptophan second limiting. And case of almonds, methionine and cysteine first limiting and second limiting, and same as nuts, lysine first limiting and threonine second limiting. Let's talk about the crude and true protein. What is true protein? True protein is the nitrogen compound only as protein, nitrogen compound found only as protein, then we can call it true protein. What about crude protein? All nitrogen compound, including also non-protein nitrogen in addition to true protein nitrogen, then we can call it crude protein. In plant samples, we can estimate soluble protein as well as crude protein. So to determine crude protein, we will use one calculation. I will show it later. And what is non-protein nitrogen? Non-protein nitrogen, that means different free nitrogen source other than true protein, like from amino acid, from ammonia, urea or biuret or nitrates. So in plant sample, if we want to estimate crude protein, then we have to multiply total nitrogen percentage with 6.25. Why 6.25? We called it Geldal constant. In Geldal method, if we find total nitrogen percentage, we can multiply it 6.25 as protein contain average 16% nitrogen. So 100 divided by 16 equals 6.25. So we will get the crude protein percent. Let's talk about the protein quality. To assess protein quality, there is 3 parameters. First one, whether or not it supply all 9 essential amino acid. First, second, this amino acid proportion and ratio of different amino acid needed for the body, and third, about the digestibility of this amino acid which is absorbed in dietary protein. On the basis of this, we can classify whether it's complete protein or incomplete protein. That means high quality or low quality protein. Various animal source which contain all these essential amino acid in correct proportion needed for the body, like milk, meat, cheese, fish, they are high quality protein, all animal protein except gelatin, because gelatin missing one essential amino acid, that is tryptophan. Another good source of protein is beef, because beef contain all essential amino acid, also iron, zinc, vitamin B12, as well as creatine and carnosine. Creatine and carnosine is very important for muscle function. What about incomplete protein or poor quality protein? As I told earlier that to have a complete protein, it must contain all 9 essential amino acid, but here lack of one or more essential amino acid from 9, and especially most plant-based proteins are considered as incomplete protein. But of course there are few sources of plant protein they can consider at high quality protein, like soy products, quinoa, buckwheat, hemp seed or chia seed. Most importantly, plant-based food don't contain enough vitamin B12. So vegans must take it as a supplement, B12 supplement, to fortify their foods. Thank you very much.